Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be explaining you all about TCA cycle. So I am going to concentrate on some of the important reactions and then the regulation of TCA cycle and applied clinical aspects related with the applied clinical aspects related with TCA cycle. Let's get into the details. Now, as you see here, TCA cycle is a final common metabolic pathways for major macronutrients in our body. So, what are the major macronutrients that we have? So, we have carbohydrates which is digested and absorbed as glucose, major carbohydrate. Then we have lipids, dietary lipids that will give us fatty acids. And then we have proteins which will be digested and absorbed as amino acids. So when glucose is undergoing oxidation in glycolysis, we get pyruvate. Pyruvate further oxidized into acetyl-CoA. So we got acetyl-CoA from glucose. Similarly, fatty acids can undergo oxidation to give acetyl-CoA. Amino acids, some of them will undergo oxidation to give acetyl-CoA, and most of them are glucogenic amino acids, but still in one or the other way they can be converted into acetyl-CoA molecules. And then ketone bodies can also be converted into acetyl-CoA, and alcohol, that is ethanol is oxidized into acetaldehyde, and acetaldehyde is oxidized into acetate, and acetate can be converted to acetyl-CoA. So that means alcohol also can be converted to acetyl-CoA. So overall, major macronutrients, ketone bodies, alcohol, all of them ultimately converted to acetyl-CoA. Note that acetyl-CoA is a 2-carbon molecule. So 2-carbon containing molecule acetyl-CoA, and these 2-carbon containing acetyl-CoA entering into TCA cycle. TCA cycle is Tricarboxylic Acid Cycle. So one of the things that you should remember here is in the TCA cycle there will be release of 2 carbon dioxide molecules. 2 carbon dioxide. In each TCA cycle there will be release of 2 carbon dioxide molecule, and the carbons of these 2 carbon dioxide actually they come from acetyl-CoA. That means acetyl-CoA here, acetyl-CoA in TCA cycle is completely oxidized into 2 carbon dioxide molecule. In return, in this oxidation process, it's going to give 3 NADH molecule and 1 FADH molecule, 1 FADH2 and 1 GTP, which is equivalent to ATP. So this is what is the product of TCA cycle. Acetyl-CoA is completely oxidized into 2 carbon dioxide, giving 3 NADH, 1 FADH and 1 GTP. And also note that if you are coming from glucose, if you are tracking carbons coming from glucose, so glucose is a 6-carbon molecule, is going to give you 2 pyruvates. Pyruvate is a 3-carbon molecule, so 2 pyruvate, that means 6 carbon. 2 pyruvates going into 2 acetyl-CoA, so there will be release of 2 carbon dioxide. So glucose carbons out of 6, 2 carbons are released as carbon dioxide from pyruvate to acetyl-CoA, decarboxylation. Now these 2 acetyl-CoAs undergoing TCA cycle 2 times, that means you are going to get 4 carbon dioxide 2 times, right? 2 carbon dioxide plus 2 carbon dioxide. Total we have 6 carbon dioxide release. 2 carbon dioxide from pyruvate dehydrogenase complex, 4 carbon dioxide from TCA cycle. So overall, TCA cycle, it is a complete oxidation of acetyl-CoA into carbon dioxide while giving energy in the form of NADH, FADH and GTP. Now let's get into individual reactions of TCA cycle. So, very first reaction is condensation of 2 carbon acetyl-CoA with 4 carbon oxaloacetate. Okay, so that is done by citrate synthase enzyme. So we get citrate. Note that citrate is a 6-carbon molecule containing 3 carboxylate groups. Okay, so that is first carbon, third carbon and the 6th carbon. I have got carboxylate group. So 3 carboxylate group. That's why this is called as tricarboxylic acid. That is why the name of the cycle is TCA, Tricarboxylic Acid Cycle, which is also sometimes called as Citric Acid Cycle or also referred as Krebs Cycle. Okay, all these three names are the same ones. Now, this is what happens in the very first reaction, condensation of acetyl-CoA with oxaloacetate to make citrate. So, acetyl-CoA carbons got into the cycle. Now, the citrate is converted into isocitrate. That is done by aconitase enzyme. So, what is the importance of this enzyme? So, this is one of the important enzyme or important reaction in TCA cycle, applied clinical aspect related with this, where citrate converted to isocitrate done by aconitase. Aconitase, it's an iron containing enzyme, and more importantly, aconitase is inhibited by fluoroacetate molecule. Fluoroacetate. This fluoroacetate is a rat poison. So, whenever rat poison is taken in by accident or for suicidal intention or whatever, so fluoroacetate is going to combine with oxaloacetate to make fluorocitrate. This fluorocitrate, it is inhibiting aconitase enzyme non-competitive mechanism. So, non-competitively fluorocitrate inhibits aconitase and totally it blocks that reaction, thereby TCA cycle don't go on. That's how energy generation in the cell will be affected. That's the mechanism of rat poison on TCA cycle. Remember, that's the applied aspect that you must be aware of. So, that's why aconitase enzyme reaction is important. Now, coming to the third reaction, number 3. Number 3 reaction in TCA cycle is conversion of isocitrate into alpha-ketoglutarate. This will be done by enzyme isocitrate dehydrogenase. So, what is the importance of this enzyme? Note that in this reaction, where isocitrate, which is a 6-carbon molecule, is converted to 5-carbon alpha-ketoglutarate. That means one of the carbon is released here as carbon dioxide. This is the first carbon dioxide that comes out of acetyl-CoA. This is how acetyl-CoA carbon is going out now. So, in this process, there is a reduction of NAD into NADH+. So, there is a formation of one NADH+, while isocitrate is decarboxylated into alpha-ketoglutarate done by isocitrate dehydrogenase enzyme. That's why this is one of the important reaction. And also note that isocitrate dehydrogenase is one of the most regulated enzyme in TCA cycle, and that you must be aware of. Okay. So, now moving on, number 4th reaction in the TCA cycle. Now, the 4th reaction in the TCA cycle is conversion of alpha-ketoglutarate into succinyl-CoA. This will be done by enzyme alpha-ketoglutarate dehydrogenase complex. That means this enzyme is very similar to pyruvate dehydrogenase complex that we have seen in our earlier lecture. Note that pyruvate dehydrogenase complex and like alpha-ketoglutarate dehydrogenase complex, it has E1, E2, E3 subunit, and it needs 5 coenzymes, that is TPP, lipoate, coenzyme A, NAD, FAD. Okay. So, with all that, alpha-ketoglutarate, which is a 5-carbon molecule, is converted into succinyl-CoA, which is 4-carbon molecule. That means one of the carbon is released as carbon dioxide, and that's the second carbon of acetyl-CoA. Because acetyl-CoA is 2-carbon molecule, so first one of the carbon is released in isocitrate dehydrogenase reaction. Now, the second carbon released as carbon dioxide in alpha-ketoglutarate dehydrogenase reaction. Okay. So, that means both the carbons of acetyl-CoA are released by this time as carbon dioxide. So, right now we have succinyl-CoA, which is 4-carbon molecule, and all those 4 carbons are actually coming from oxaloacetate. Remember, in the very first reaction, oxaloacetate condenses with acetyl-CoA, 2-carbon condenses with 4-carbon oxaloacetate, releasing 2 carbons as carbon dioxide. Those carbons are from acetyl-CoA. Now, the succinyl-CoA carbons are actually from oxaloacetate. Remember that. Now, what are the important points about alpha-ketoglutarate dehydrogenase complex enzyme? Number 4th reaction in TCA cycle. So, some of the points here is arsenate. Arsenate is going to inhibit this reaction by binding to E2 subunit lipoic acid, which has got thiol group, just like what we have seen in pyruvate dehydrogenase complex. So, arsenate inhibits this reaction. That's the important point there. Now, the second point is carbon dioxide, second carbon of acetyl-CoA released here. So, it's a decarboxylation reaction. Third point is there is formation of NADH plus in this reaction. So, and also alpha ketoglutarate dehydrogenase complex, it needs 5 coenzymes for optimal function. What are the 5 coenzymes? TPP, lipoic acid, coenzyme A, that is pantothenate derivative, NAD, niacin derivative, FAD, riboflavin derivative. Okay, deficiency of any of these will lead to decreased activity of alpha ketoglutarate dehydrogenase complex. Arsenate inhibits this enzyme. Remember that also. So, you are going to get one NADH plus here, and if that NADH plus goes into electron transport chain, it is 2.5 ATP, worth 2.5 ATP. Every NADH plus is worth 2.5 ATP, especially if they are made in the mitochondrial matrix. Remember that point. So, these are the important points related with this particular reaction, and also this reaction is regulated, one of the regulated enzyme here. Okay, coming to number 5 reaction. So, we have finished number 1, number 2, number 3, number 4 reaction. Number 5 reaction, 5th reaction in TCA cycle is conversion of succinyl-CoA, 4-carbon succinyl-CoA into 4-carbon succinate. Okay, so here the reaction is done by succinate thiokinase enzyme, where CoA is removed, high energy thioester bond is removed here, and during this process GTP is made. That means GDP is added with PI, inorganic phosphate, to make GTP. So, there is a generation of high energy phosphate in this reaction, so giving GTP, which is equivalent to ATP. So, this is a phosphorylation going on in the matrix of mitochondria without going into electron transport chain. So, this is an example for substrate level phosphorylation. So, there is only one example for substrate level phosphorylation in TCA cycle, and that is conversion of succinyl-CoA into succinate done by succinate thiokinase enzyme. And also, wherever there is a need for GTP in our cells, one of the source for that GTP is succinate thiokinase reaction in TCA cycle. Remember that. That is the importance of this reaction. That's about number 5th reaction in TCA cycle. Now coming to number 6 reaction in TCA cycle, 6th reaction. 6th reaction is conversion of succinate into fumarate done by succinate dehydrogenase enzyme. And this reaction, FAD is converted to FAD2H, which is 1.5 ATPs if it goes to electron transport chain. Okay, so what is the speciality of this enzyme? Unlike all other TCA cycle enzymes, succinate dehydrogenase enzyme, it is embedded in the inner mitochondrial membrane, and in fact it is complex 2 in the electron transport chain. So, it is complex 2 in the ETC, electron transport chain. That means succinate dehydrogenase not only participate in TCA cycle, it is also part of electron transport chain, where FAD is going into FADH to FADH2, entering into electron transport chain and give 1.5 ATP. So, FAD is a derivative of riboflavin, that means riboflavin deficiency may affect this reaction in TCA cycle. Remember that. So, those are the applied things related with this reaction. Coming to conversion of fumarate into malate done by fumarase enzyme, it's a nitrolase enzyme, so not really that applied clinical aspect related with this enzyme. Coming to conversion of malate to oxaloacetate. So, this reaction is catalyzed by malate dehydrogenase enzyme, where NAD is converted to NADH plus H plus molecule, and this NADH plus is worth 2.5 ATP. So, ultimately what happened in the TCA cycle is this 4-carbon oxaloacetate, it is regenerated at the end of TCA cycle. So, what is actually consumed is acetyl-CoA, which is releasing 2 carbon dioxide molecules. So, there is complete oxidation of acetyl-CoA, while oxaloacetate is regenerated at the end of every TCA cycle. So, oxaloacetate carbons are not really consumed. It is the carbons of acetyl-CoA which are basically released here as carbon dioxide. That's what is all about TCA cycle. So, that means 2 carbon dioxides are released for every acetyl-CoA being oxidized, and then NADH, 3 NADH molecules in fact are made, and each NADH is worth 2.5 ATPs in the electron transport chain. One FADH₂ is made which is worth 1.5 ATP, and one GTP is made which is equivalent to 1 ATP. So what are all the reactions that make NADH plus in the TCA cycle? And that is isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, and also malate dehydrogenase. So which enzyme makes FADH₂ in the TCA cycle? It is succinate dehydrogenase. Which enzyme makes GTP in the TCA cycle? It is succinate thiokinase, and it is an example for substrate level phosphorylation. Remember that. Okay, so let's calculate number of ATPs that you get from oxidation of acetyl-CoA. So when acetyl-CoA is oxidized into 2 carbon dioxide molecule, we're going to get 3 NADH plus 3H plus molecule. So that means each NADH is 2.5. So 2.5 times 3, that will be 7.5 ATP. And then you get one FADH₂, and each FADH is 1.5. So I will add 1.5 here. And you get one GTP, and one GTP is equivalent to 1 ATP. So I will add 1 here. Okay. So that means we get total 10 ATPs. 10 ATPs per oxidation of acetyl-CoA molecule. Okay, so that's what is the energetics of TCA cycle, which I have shown here. So each acetyl-CoA molecule will give 10 ATP. Okay, so let's move on to see regulation of TCA cycle. So as I told you, most regulated enzyme in TCA cycle is isocitrate dehydrogenase. So whenever there is a lack of energy in the cell, so that means there will be more ADPs in the matrix of mitochondria. So elevated ADP simply indicates more ATPs need to be generated. So what this ADP will do? ADP will have a positive allosteric effect on the isocitrate dehydrogenase enzyme, thereby increases the pace of TCA cycle. TCA cycle is stimulated and it is going to oxidize more and more acetyl-CoA into carbon dioxide, thereby making more and more NADH, and that NADH can go into electron transport chain to give more ATPs. And also when the person is exercising, when there is a increased levels of calcium in the matrix of mitochondria, calcium will have a positive effect on isocitrate dehydrogenase and keep this cycle, TCA cycle, active. That's why when person is exercising, so there will be stimulation of isocitrate dehydrogenase enzyme, thereby TCA cycle runs much faster. So lack of energy means more ADPs and more calcium, especially which is seen under muscle contraction whenever person is working out. So TCA cycle need to be activated so that oxidation of glucose and fatty acids need to be done. So acetyl-CoA is oxidized to give more and more energy. That's how positive effect will go on. So calcium also will have a positive effect on alpha-ketoglutarate dehydrogenase enzyme complex. So these are the two positive modulators on isocitrate dehydrogenase and one positive modulator on alpha-ketoglutarate dehydrogenase complex. And now the negative modulators of TCA cycle is elevated levels of NADH. So whenever a person is in fed condition and resting, so NADH levels will be more, and higher NADH levels means there is no need to run TCA cycle faster because there is no need of energy. So higher NADH will have a negative effect on isocitrate dehydrogenase reaction, alpha-ketoglutarate dehydrogenase reaction, and also malate dehydrogenase reaction. So three enzymes are negatively modulated by higher levels of NADH plus in TCA cycle. Now one of the regulation on TCA cycle can also be on the very first enzyme, that is citrate synthase enzyme, where citrate will have a negative effect, feedback negative effect on citrate synthase enzyme. So like this, TCA cycle is regulated at various level, and most important reaction where TCA cycle is regulated is isocitrate dehydrogenase. Remember that. Now let's calculate ATPs that are coming from complete oxidation of glucose. So when glucose is undergoing glycolysis in the cytoplasm, so you get 2 pyruvate. One glucose will give 2 pyruvate, and net ATP yield from a glucose into 2 pyruvate under aerobic condition with malate aspartate shuttle mechanism, it is 7 ATP with MAS, malate aspartate shuttle mechanism. If it is a glycerol phosphate shuttle mechanism, I will write it as GLY3 phosphate, you are going to get 5 ATP. So either 5 or 7. That makes a difference here because shuttle mechanism, NADH plus from the cytoplasm, how it is carried into the matrix of mitochondria, that makes a difference of 2 ATPs. So either 7 or 5 coming from glucose to 2 pyruvate. So let's keep it that way. And 2 pyruvates into 2 acetyl-CoA. 2 pyruvate into 2 acetyl-CoA, we get 2 NADH, and these 2 NADH is worth 5 ATP. So that is 5 ATP there. And of course you get 2 carbon dioxide. Okay, so 5 ATP we got from there. 2 pyruvate to 2 acetyl-CoA. 2 acetyl-CoA into 2 times TCA cycle. Each acetyl-CoA is 10 ATP. So 10 ATP plus 10 ATP. So total we have 20 ATP there. So with the malate aspartate shuttle mechanism, we have 5 ATPs, 2 plus 5, that is 7 ATPs there. 7 ATP, net 7 ATP from glucose to 2 pyruvate, and pyruvate to acetyl-CoA, 5 ATPs, 2 pyruvate to 2 acetyl-CoA, and 2 acetyl-CoA in the TCA cycle will give 20 ATP. So that means 7 plus 5 plus 20. That means we get 32 ATPs using malate aspartate shuttle mechanism. If it is a glycerol phosphate shuttle mechanism, this 7 changes to 5 there. Okay, that will be 5 plus 5 plus 20. That will be 30 ATPs in glycerol phosphate shuttle mechanism. Okay, so this is how TCA cycle can give energy from complete oxidation of glucose. Now, note that TCA cycle, it's an amphibolic cycle. Amphibolic cycle means it is catabolic and also it is anabolic. So it is a combination of catabolic and anabolic depending on the needs of the tissue, needs of the cell. It can be totally catabolic where acetyl-CoA is completely oxidized into carbon dioxide, or some of the intermediates of the TCA cycle, it can be taken out of TCA cycle to synthesize something like anabolic process. That's why TCA cycle is referred as open cycle. Okay, now let's see what all the things that can be taken out of TCA cycle to make new molecules. Like citrate, whenever person is in fed condition, so citrate can build up because NADH accumulates, it will have a negative effect on isocitrate dehydrogenase and other 2 enzymes that we have seen. And so when citrate builds up, that citrate will be moved out into the cytoplasm and be converted to fatty acid. It can go into cholesterol synthesis. So that's how TCA cycle intermediate citrate can be diverted into fatty acid and cholesterol biosynthesis. Or alpha ketoglutarate, which is a TCA cycle intermediate, can be converted to glutamate, and glutamate in turn is converted to gamma aminobutyric acid, which is a neurotransmitter. That's how it can go into neurotransmitter synthesis also. Or succinyl-CoA. Now succinyl-CoA is one of the TCA cycle intermediate. So especially in the hematopoietic tissues or any tissue where heme synthesis is needed, so succinyl-CoA can be taken out of TCA cycle and it can be placed into heme synthesis in the mitochondria. Okay, so that's how succinyl-CoA can go into heme synthesis. So basically succinyl-CoA, it condenses with glycine to make delta aminolevulinic acid, and ultimately delta aminolevulinic acid can go into heme synthesis. Or when the person is in fasting condition, the malate, which is part of TCA cycle, it can be taken out of mitochondria into the cytoplasm and it can be placed into gluconeogenesis, like new glucose formation, especially in the liver. So malate can go out into gluconeogenesis process. Or oxaloacetate, which is part of TCA cycle here. oxaloacetate can undergo transamination reaction to make aspartate. So that means a non-essential amino acid aspartate can be synthesized using oxaloacetate, which is a TCA cycle intermediate. So like this, TCA cycle is an open cycle. So depending on the needs of the cell, it can be getting out of the pathway to go into some other metabolic needs of the cell. Okay. Now, whenever there is a TCA cycle intermediate is taken out of it, so there will be gap that is made. Let me show you that here. So if I am taking oxaloacetate out of the TCA cycle to make aspartate, so there will be gap that is created here because oxaloacetate is taken out, aspartate is made. But to continue TCA cycle, you need oxaloacetate because oxaloacetate condenses with acetyl-CoA in the very first reaction to make citrate. So during that time, there are some gap filling reactions that will be present in our cells, and these gap filling reactions are referred as anaplerotic reactions. Basically, they will replenish a specific TCA cycle intermediates that are taken out of the TCA cycle. So in order to continue TCA cycle, so gap filling need to be done. So one of the enzyme that we study here in a gap filling or anaplerotic reaction is pyruvate carboxylase reaction. So pyruvate carboxylase, what it does, it's going to convert pyruvate, which is a 3-carbon molecule, into oxaloacetate, which is 4-carbon molecule. Okay, so 3 carbon converted to 4 carbon. So that means there is carboxylation process. That means carbon dioxide is getting into the reaction in the form of bicarbonate, and it's a condensation reaction. That's why you need ATP. Okay, so and as you know, most of the carboxylases, they need biotin as the coenzyme. So pyruvate carboxylase, it needs carbon dioxide, it needs ATP, and it also needs biotin. So ATP, biotin, carbon dioxide, A, B and C, ABC. It needs A, B, C. Pyruvate carboxylase needs A, B, C, ATP, biotin, carbon dioxide. And also pyruvate carboxylase, it needs an obligate activator, that is acetyl-CoA. Only in the presence of acetyl-CoA, pyruvate carboxylase will work. It's an obligate activator. Remember this, obligate. Obligate activator means without the presence of acetyl-CoA, pyruvate carboxylase do not work. There are only two enzymes in our body that needs obligate activator. One is acetyl-CoA, which is necessary for pyruvate carboxylase. Other is carbamyl phosphate synthetase 1 enzyme in the urea cycle that needs N-acetyl glutamate as an obligate activator. So there are only two enzymes that needs obligate activator in our body. One such enzyme is pyruvate carboxylase. Obligate activator is acetyl-CoA. Remember that. Now, this slide is showing you other gap filling reactions other than pyruvate carboxylase reaction, which I have shown here. Pyruvate to oxaloacetate done by pyruvate carboxylase. Like that. So if there is a glutamate taken out of the TCA cycle, so most of the amino acids can be converted to glutamate. So glutamate can undergo oxidative deamination process to make alpha-ketoglutarate, and or it can undergo transamination reaction to make alpha-ketoglutarate. That's how it can be replenished. Or if the succinyl-CoA, if it is taken out of TCA cycle for heme synthesis, at that time, so if succinyl-CoA is taken out of TCA cycle, at that time the succinyl-CoA can come from catabolism of valine, isoleucine, methionine, threonine and all that, where it will give propionyl-CoA, or even odd chain fatty acids oxidation will also give propionyl-CoA, which is a 3-carbon molecule, and 3-carbon propionyl-CoA can be converted to 4-carbon succinyl-CoA by propionyl-CoA carboxylase and methylmalonyl-CoA mutase enzyme. So like this, a gap filling can be done whenever there is a gap that is created in the TCA cycle. So this is all about a TCA cycle, reactions, regulation and applied clinical aspects. Thank you.